Hi friends, welcome back to another section of immunosuppressants. It's part 2, and here I am depicting you as I told you in the previous video section that I am representing two cells over here. One is T helper cells and the other one is APC cells, and APC cells are called as antigen presenting complex cells and it is called as T cell. As I said here, you have different type of cell complexes over on top of APC, that is called as MHC, which is called as multiple histological complex, and also onto the T cells you have different types of proteins which are called as CD4, nothing but called as cell of differentiation, and also you have CD3, which is called as cell of differentiation or cell of designation 3, and also onto the second center one you have T cell receptor. Here what it is T cell receptor, where the complex will come and attach over here. This is called T cell receptor. Okay, so once the APC will come and sit onto the T cell, the complex will start to attach itself, making some conformational changes, and the antigen will be inside and engulfed inside the T cell. And this T cell, once it enters into it is attached to a complex called as calmodin. Here I gave you the word here. This one, which is been over here, I'm doing it one more with a black pen. This is called as calmodin. So what is calmodin? Calmodin is a protein molecule which is a complex of calcium ions present over here. So the one antigen presenting cell will enters into the T cell with the help of receptor. It starts acting onto the calmodin type of cell, where it is being taken up into two parts. One is the Ca2+, nothing but the calcium ions are being channelized, and also it is releasing some CAM. What is CAM? CAM is calmodin activated membrane complex, which has been eliminated to it. So from the calmodin complex or the calmodin protein molecule, you can see two particles coming outside. One is calcium and the other one is CAM. The CAM is called as calmodin activated membrane gene. So what is help of calcium here? The calcium is used to further depolarization or the hyperpolarization of the T cells, so that it is being action potential and the other works will be working over there. And what is CAM? CAM is nothing but the calmodin activated membrane channel, which has been a complex which is used for the production of cytokines. And so this is a complex which is being released from the calmodin, and after entering outside it is being differentiated into two parts. One is calcium and the other one is CAM. As I said before, this calcium is used for the depolarization. That means action potential, the firing of the T cells will be useful. And what is CAM? The CAM is useful for the production of cytokines here, and how the cytokines are produced. And once the CAM is being produced, it is been releasing one more factor, what is called as protein kinase C. And this protein kinase C is very important for the IP3 transportation or the calcium action potential transportations by two pathways. One is IP3 and DAG, but actually it is not important over here, but just it's a word how the calcium has been synthesized or how the calcium is being polarized inside the action potential. And it is being done by the protein kinase C. And what is protein kinase C? Protein kinase C is one of the released products from the CAM, and these are the useful for the production of IL-1 and IL-2. What are these IL-1? IL-1 is interleukin and IL-2 is interleukin 2. So these are also called, when we call as cytokines. What are these all three? These are all called as inflammatory factors. Inside, what is the result of AG and AB complex? It is nothing but the inflammation. It is giving the signal for the body to fight against. So it is a symptomized signal, that is called inflammation. What it is releasing? This PKC, nothing but protein kinase C, is releasing some inflammatory factors, what is called as IL-1 and IL-2 and cytokines, so that the inflammation factors are released out and the production of antigen and antibody complex results comes out in the form of inflammation. So the body will be alarmed whether it is done by the inflammation or whether it is done by the fever or any other symptoms, that something is inside your body which is not favor for your condition of your body. It may get into the diseased parts. It is symptomized condition. And later, this calmodin factor is not only releasing some calcium and CAM, but it is also releasing a factor. What is that factor? This is called nucleus factor activating transaminase factor. It is one of the factor of genes which are being released by this calmodin section, which is developing or acting on the cytokine gene. And what is this cytokine gene? Cytokine gene is nothing but the inflammatory gene which are used for the production of IL-1, IL-2 and I cytokines 1, 2 and 3. So what you can understand from this is whenever the AP cell has been stick onto the T cell with the taking up the receptor cell, it enters into the T cell, and what it is doing some different type of actions over there. One is it is acting on the calmodin gene. What is this calmodin? The calmodin is nothing but a small component which consists of calcium ions over it. It is a house of calcium, we can say. And it is also not only with the calcium ions, but it is also having one more enzyme called as CAM. What is CAM? As I told you, it is called calmodin activated membrane enzyme. And so whatever this complex has been released out, and after complex has been released out, it is differentiated into two parts. One is calcium and the other one is CAM. So what is calcium? Calcium is used for the hypolarization and the depolarization of the action potential, and the firing of T cell will be happening over here. And the CAM, what is CAM? CAM is nothing but using for the production of PKC. What is PKC? It is called as protein kinase C. And this protein kinase C is one of the genes which are being released from the CAM. What is this PKC is doing? It is doing the pathways for the production of inflammatory factors. What are those inflammatory factors? These are IL-1, IL-2 and different type of cytokines. And not only it is releasing cytokines, it can be taken into consideration what is called as TNF-alpha. Nothing but tumor necrosis factor alpha. And this from the calmodin, another factor is also being released. What is that called as NAFT? NAFT, what is NAFT? Nuclear factor activating transamination enzyme. And this transaminase enzyme is being acting or stimulating one of the genes which are present in the C cells, and that gene is called as cytokine gene. What this is doing? This cytokine gene is directly producing IL-1, IL-2 cytokines and TNF-alpha. So what we can see is that pathway may be this type of pathway which is producing the inflammatory factors, or it may be releasing your nucleus factor which is being acting on the cytokine gene and also making the inflammatory factors. So there are two types of pathways which are being done inside the T cell so that the symptomized of the inflammatory factors will be released. And if at all the fever has to be symptomized, here you can see the consideration of PG also. And this PG will be came into the picture once it enters into brain. The thalamus region, the hypothalamus region, where the inflammatory factors of PGE will be, because it is called as thermoregulator in the brain. So whenever this symptomized, this inflammatory factors will drive out to the brain via the circulatory system. It will start acting onto the hypothalamus thermoregulatory system, where it is been releasing PGE factors, nothing but the prostaglandin factors. And once these prostaglandin factors are being synthesized over there, you will get fever. That means the body is being symptomized here with the inflammation and here with the fever. So once these two, all the factors are being eliminated outside, so that the immunity system will be taken out. So actually this is called as immunity cycle which is being done that. But coming to the topic what we have started here, this is called as immunosuppressants. So whatever the cycle over here being circulated over here has to be inhibited with some type of drugs, so that this circulatory system will be stopped up. Just I'll give you the centers where we can stop off those immunosuppressions. One is, this is the area where we can control the immunity system. The other one is the TTD4. This is called the second factor, where we can act some drug so that the recognition is not being happened. And also we can start inhibiting over here, that means the calmodin, where the CA and CAM will not release. And also we can control this area where the differentiation of calcium and CAM had been done. This is a fixed factor, and we can see the next factor is inflammatory factors where had been symptomized with their type of inflammatory factors, and this is a fifth place where we can stop off at starting mechanism of action. And also we can see here one more protein molecules are given over here. What it is called as cyclophilin. What is cyclophilin? Cyclophilin is a type of protein where the drugs are being attached, and that part is attached to the calmodin part, so that the suppression of the T cells will be happening over here. So we can see the 6 spots over here where the suppressants can be acted and the suppression of all the immunosuppressants will be taken. So the first factor will be the macrophageal factor where the suppression with some drugs will be taken out. The second place is CD4 where the recognition is made not done, and the complex between MH3 and CD4 is been not done. And the third place is the calmodin protein, which is being a house of calcium and CAM. This can be inhibited or blocked off. And the fourth center is the differentiation between the calcium and CAM, so that they are not being released from the CAM complex or the calcium complex calmodin. And the fourth place is the differentiation between these two. And the fifth place is the symptomized of eliminatory factors. That means the inflammatory factors which are being released. That means they are acting on IL-1, IL-2, cytokines and TNF-alpha. And they are also not only acting on these places, they are also acting on the cyclophilin protein which is present inside the drug attached to the cyclophilin part, and that protein is called as cyclophilin protein. And the remaining classification and the mechanism of actions will be done in the next section. Thank you for the class.